Good morning, everyone, and happy St. Patty's Day. My name is John Heering, and welcome to the Tribe Public presentation and Q&A webinar event titled Keys to the Kingdom, Unlocking the Power of Your Immune System, featuring Indaptus Therapeutics CEO Jeffrey Meckler. As many of you know, I am the managing member of Tribe Public. Our website is tribepublic.com. That's TRIBE public.com. Members from 31 plus countries in all 50 states have joined the Tribe Public, or joined the tribe, as we say, freely to gain direct corporate access to leaders and experts that they care about. Our webinars, via our webinars, and also via our 36 venue sites located across the U.S., where we host in-person CEO events, luncheons, dinners, speaking engagements, with local tribe members. Tribe members furthermore submit names of experts and leaders of industry via our free wish list process at our website, adding names of companies and subjects that span across all sectors so that they can learn more. Please note that I'm also the managing director of Vista Partners, LLC, a registered investment advisor in California, and its website is vistapglobal.com, VISTAP as in Paul, global.com. Please review both sets of disclaimers at each site and know that currently I'm a shareholder and an advisor to Indaptus, our presenter today. It trades under the NASDAQ symbol INDP, Indigo Nancy David Paul, INDP. I would also like to thank all of you for participating and for all of your questions that you have already submitted, and remind all that they can send in additional questions via the Zoom chat feature during this event. We will do our best to get them addressed during this approximate 30-minute event. We will also endeavor to publish the event video of this event today at our Tribe Public YouTube channel at some point later today, and you can view anything that you may have missed and or pass that on to others that may have not been able to attend today at this time. Thank you again, Indaptus and Jeffrey Meckler, for joining us today. Now let's get started. Jeff, to begin, as there's some new folks on here, and obviously we'll see in your video in the future, can you tell us a little bit about yourself and what brought you to being the CEO of Indaptus today?
Certainly. And first off, happy St. Patty's Day to you and everybody else. Hopefully there's some green beer in my future this evening. My background's pretty simple. I came out of graduate school and joined Pfizer, spent a little less than 2 decades there, where I worked in a whole variety of groups, from manufacturing to sales ops to support to market research to a whole bunch of things. Ended up doing business development. Left that and went to work for some different healthcare private funds. And that kind of just led me into being involved with the management of some of these companies, being on some boards, and eventually being CEO of several small companies. And we formed Indaptus about 3.5 years ago and kind of started down this journey. And with that, let me kind of jump into Indaptus. We're a unique company. And the slide everyone's seeing, I hope you all can see it. This is a picture of an E. coli. It's a gram-negative bacteria. This is the starting point, the core basis of our technology. And I'll describe a little bit more in a few minutes how that works. But this is what we start with. And it's a naturally based approach. And essentially, the idea came out of the concept that we see and by the way, it's been documented for hundreds of years. We see out there in the world that tumors have contracted, regressed in the presence of a bacteria infection. So we learned in the last few decades that bacteria have all of these toll-like receptors, all of these immune agonists. So our goal was to figure out how we could adjust what we see in nature to safely give it to humans so that you don't give a bacteria infection, but at the same time, get that immune activation going. Now, just so people know, I will be making forward-looking statements, and please check our filings for the latest information. So this is the highlights slide. So if there's one slide to kind of understand what we do, this is it. We are a clinical company. We started this company 3.5 years ago. In that time, we filed our IND, which is the investigational new drug application with the FDA. We got that cleared. We made GMP, which is the material you need to inject into humans. We made that, and then we started this clinical trial. And what you see in that second set of bullet points, we completed our first cohort in 2023, the second cohort in 2024. We initiated multi-dose, which is our weekly, in the middle of 24. And then last fall, we actually were able to sign an agreement with BeiGene. BeiGene is a $25 to $30 billion oncology company that has a drug called tislelizumab. It's an anti-PD-1. And in our preclinical studies, our therapy combined with a PD-1 inhibitor shows the greatest activity. And so we're delighted to sign this agreement, and we intend to, in the near future, launch that study. And we just recently announced last week that we had completed more than 20 patients in the weekly dosing. And some of those patients were demonstrating stable disease. Now, stable disease basically means the tumors really aren't growing much. So we're seeing some activity. We'd like to see more, but we think in that combination study is our first kind of foray into looking for that type of demonstration. We've also been presenting a lot of this information at major conferences like AACR, ASCO, SITC. And we've been presenting to that. We presented the latest data in the last quarter at the Society of Immunotherapy. But the questions I always get from investors are, what's next? What are you going to do next? The big thing for us is this combination study with BeiGene's tislelizumab. That to us is a real pivot point for the company, and it's really a pivot point for us to understand more about the technology. But over the course of the year, not only will we start that study, but we'll have readouts on the safety and efficacy of this weekly dosing. And what we hope to do is sometime later this year or early next year, really get an insight into the proof of concept. Can we reduce tumor, get some tumor regression in this combination? The other thing I should mention is that this technology actually has application beyond oncology. This is our first foray into using the technology, but there are applications in antivirals and in vaccines, anywhere where immune modulation could have a benefit to the therapy. So what's the problem we're trying to solve here? Well, it's really simple. Over the course of the last few decades, immunotherapy in cancer has made huge strides. But what we've always come down to is this belief that if you do too much to the immune system, it will over-rev itself and attack itself. And this comes from the fact that we've seen things like cytokine storm or macrophage activation syndrome. These are components where you kick up a number of pathways in the immune system, and the immune system gets out of control. That mentality has led us to this approach where we only try to do 1 or 2 switches in the immune system. But you have to understand, our immune system is very complex. And what goes on naturally in cancer is that cancer is simply cell programming gone awry. And when the cell programming goes awry, it then forms tumors and keeps growing. And the immune system just doesn't recognize that these cells shouldn't be here. What happens naturally is when this is occurring and the systems are working right, different cells from both the innate pathways and the adaptive pathways will attack these cancer cells. As the cancer grows, this is not happening. Very often, it could be one switch, one component can be the tipping point. That's why checkpoint therapies or targeted antibodies work so well, but they only work in a small population of patients. Our concept is, let's take a look and have a package of immune activators so we engage the entire immune system. The challenge has always been how to do this safely. That's where the Decoy technology and this gram-negative bacteria come into the picture. So as I said, current immunotherapies, they target only one or a few components of the immune system. The other thing these therapies do is they are in the body for long periods of time. If you look at checkpoint therapies, they're usually dosed every 21 days. That means for weeks on end, you have this therapy hitting your immune system. That's why they can only hit one or two switches. This long duration, from literally weeks to months, is what really leads to the limiting toxicities of dosing. So really what's going on is you have this narrow therapy for a long period of time. And doing so, still, we see survival rates, 5-year survival less than 20% in advanced cancers. What we're doing is flipping that idea on its head. And we're saying, let's get a broad package of immune agonists and give it in a short period of time. We call this the pulse prime activation. It originally was our pulse prime hypothesis. We proved it as we dosed the first few cohorts in this study. What we've seen by doing this is an activation that transiently activates more than 50 cytokines and chemokines. And these may work synergistically to target the tumor. So the next question always is, what the heck is this? So this is a gram-negative bacteria-based therapy. We start with gram-negative bacteria. And the reason we do that is we've evolved as humans with gram-negative bacteria. In fact, gram-negative bacteria has all these innate and adaptive immune activators naturally existing. One of the things that's a piece of trivia is you have as many bacteria cells, if not more, than you have human cells in your body. We evolved with bacteria, and that has kind of set up this relationship of how we identify the bacteria in our immune system. Now, the bacteria also gets excreted out of the body very quickly. The liver and spleen pick up the bacteria in a very short amount of time, mostly in less than 2 hours. So what we're getting with gram-negative bacteria is not only a package of naturally existing immune activators, but also the short duration. And that's why we think having this package, the innate and the adaptive, in a very short pulse is what will really help the immune system do what it's designed to do, reset and identify the cancer and regress it. So we take a gram-negative bacteria, it's engineered so they can't replicate in humans. It requires a molecule that humans do not have. We then deactivate what is called lipopolysaccharide. This is a component of the outer membrane. The reason we do this is LPS is the most potent immune activator. It limits the amount of therapy we can give. But what the real genius behind this technology is looking at the ratios. What we do is 75% of the bacteria membrane is made of LPS. By reducing it roughly 90%, it flips the ratios of all the other immune agonists. Think of it this way. You're in a recording studio and you're at this mixing box and you turn down the synthesizer. When you do that, you still hear a little bit of the synthesizer, but now the drums, the guitar, the bass, you can hear the rest of the band. That's what we're doing, is we're really changing these ratios in a way that really allows a broader and more efficient activation of our immune system. Once we've done that, we kill the bacteria and we cross-link the outer membrane. We just make it stable so it gets picked up by the body's immune systems better. Then we use this killed, stabilized bacteria and we put it into an IV bag, and we give an IV. It's about a 1-hour infusion. That's the decoy therapy. So as I mentioned, the exciting part about 2025 is as we're in the first half of the year, we are at the point where we're getting close to initiating our combination study. Now, if you go back and look when we formulated this company 3.5 years ago, you will see we are relatively on track to do what we said we were going to do. That's one of the things I'm most proud about the Indaptus team is we will execute. I can't promise you what the results of the study will be, but I can tell you we will do the science and we'll do it in a way that answers the question, do these decoy therapeutics add to synergistically to PD-1s? PD-1s only work in 1 out of 4, 1 out of 5 patients. If we can increase that, that is a huge outcome to solid tumor cancers. So on the left side, you've seen a number of our key milestones over the past year or so. But more excitingly, what's going to happen this year? We're going to initiate our combo study. We're going to have more data on the safety of the weekly dosing. We'll get some efficacy data on the weekly dosing. And then we'll also get some proof of concept data. As we dose more patients in the combo study, now remember, it's an open label study. We don't have to wait to the end and then unblind it like a placebo-controlled study. We are open label, so we'll see it as the year goes on. Either sometime later this year or next year, we'll really have a sense of that proof of concept data. So what have we observed in humans so far? Well, we basically have proven our hypothesis. Decoy 20 clears within 2 hours. What we see is this transient, this pulse of more than 50 cytokines and chemokines. And these are all different cytokines and chemokines involved in anti-tumor responses. We've seen the tolerability very consistent. And what that means is we see the side effects are mostly mild to moderate. And the side effects are you feel like you have a cold or a flu, some sort of bug, because that's what you feel like when your immune system kicks in. You know, I think the technical term is malaise, but you're tired. You have, achiness, maybe a little nausea, maybe a little fever. This is what you feel like during the infusion when we give Decoy 20. These things like fevers and chills, they all are transient and they resolve within 24 hours to 48 hours. That's the exciting part, is that it is activating the immune system and we see it acting like we expect it. Now, I encourage everyone to go to our website. We have lots of data and lots of posters from scientific presentations. I'm just showing you one example of one poster, but it illustrates what happens. What you see here is TNF alpha. This we know has anti-tumor activity. And what you see are these are 4 patients that were dosed a single dose. And what happens is this spike occurs initially at dosing the decoy therapeutic. And then over the course of 24 to 48 hours, it comes back down to baseline. This is what our therapy is doing. Our belief is by doing this multiple times, and by the way, in previous iterations and approaches of doing similar type of, whether it's bacteria or virus immune mediation, this pulse has been shown to have some impact where you're resetting the immune system. You're doing this, but it's not hitting the immune system so hard that you have all the side effects of cytokine storm, for example. So why are we different than all these approved therapies? So really, CTLA-4, anti-PD-1, CAR T therapies, they're really focused on how we can activate particular T cells that we know will attack the cancer. Decoy Therapeutics, by being this broad, innate, and adaptive, also does the macrophages, the dendritic cells, the natural killer cells. And so this slide is really indicative and identifying those changes that occur between us and other therapies. We are much broader in a shorter duration. These are really narrow. Now, an example of that is also here in showing you toll-like receptors, individual ones, and a decoy package of immune agonists. So here you see a TLR9, a TLR3, a TLR4, and then this package of decoy 10, which is a 2, 4, 8, and 9 package. And what you see is that these are different anti-tumor cytokines. So these are the precursors that tell the immune system which cells to go after. It's a simplified way, but it's, these cytokines help engage the immune system. And what you see is individual agonists don't work as well as a package. There's a synergy of these agonists, and it's not just a synergy of adding on, it's they work together. And we found a way to do that by harvesting them off of the gram-negative bacteria. So what does all this mean? So in preclinical models, when we take a decoy therapeutic and add a checkpoint inhibitor like an anti-PD-1, and this is in hepatocellular carcinoma, we give these mice a liver cancer. You can see the top blue line is the control. And essentially, these are the mice we do nothing with but give them the cancer tumor, and the tumors grow. If we give the mice a anti-PD-1, you'll see the tumor grows, but it doesn't grow as quick. It's kind of delayed a little bit. And 1 out of 6 of the mice has a complete response. That means the tumor shrank to 0. Now, if we give a decoy therapeutic, we see 2 out of 6 have a complete response. And the line is essentially similar to the anti-PD-1. So basically, in mice, the decoy therapeutic and the anti-PD-1 have a similar tumor regression curve. The real excitement for us is really around this combination, because when you add the decoy and the anti-PD-1, you get a 6 out of 6 complete response. And one of the things, if you compare us to other companies and you look at these slides, you'll notice we don't go just 28 days. We're going out 70, 90, 120 days, because we really want to understand, is the immune system doing what it's designed to do? This is what we're trying to recreate here with our combination study in humans later this year. Now, I will preface this, we do not expect 100 or 80% complete responses in humans. Mice and humans are different. There's a lot of things you need to kind of understand. But our goal is, if we can go from 1 out of 4 or 1 out of 5 to 2 out of 4 or 2 out of 5, double the response rate in humans, this is just a game changer. But then there's something else that goes on with our therapy. And here we go. So here's what we see when we re-challenge. So this is a different study, different mice. 12 mice given a decoy therapeutic. 11 of the 12 had complete regression. That means they were tumor-free at 90 days. You see that one mouse, I can't explain. I always get asked, why did that mouse not work? I do not know. But 11 out of 12 mice had a complete response. And this was the same regimen as on the last slide, hepatocellular carcinoma with a decoy therapeutic and an anti-PD-1. But on the 91st day, we re-challenge. We give them another hepatocellular carcinoma tumor, and we see what happens. And the tumors grow a little bit. But as the tumors are growing, then the body realizes, wait a second, we just saw this before. We know what to do. So what they do is they attack the tumor. 100% of the mice who were a complete response actually regress and are another complete response. And we call this immunological memory because we gave them no therapy. This is your body now realizing this is a cancer. It understands the antigens. It knows what it needs to do to remove that. And that's the real key. This is the holy grail of immunotherapy, when the body learns how to deal with this in the future. Same concept when we give vaccines. We're teaching the body, hey, when you see this, this is what you do. So one of the things we want to understand is how that might be the case. And this is and we have many of these on our website, by the way, but this is a gene expression analysis. Essentially, blue means the gene doesn't turn on, red means the gene is on. And these are genes that we know have immune pathways that for tumor regression. And what we see is we did every combination. By the way, the mice also received an NSAID. Patients in our trial receive an NSAID as well. That's to alleviate some of the fever and discomfort. But what we know is with the NSAID, with the Decoy, and with the anti-PD-1, we basically take these tumors. All these genes are blue. They go to red. That means they're activated. This helps explain why we see that 80 to 100% complete response. So just kind of stepping back, I know I gave a lot of heavy science there. I tried to keep it light, but the whole point of all of this work is that advanced cancers still have a low percentage success rate out, and you see 3 to 17% 5-year survival for patients with metastatic disease. The ones that are in blue are some of the initial potential types that we're looking at. There's just great opportunity here, both from how we change the paradigm of cancer treatment, but also from the business standpoint of where is their market potential. As I mentioned before, this has application outside of oncology. There's both antiviral and vaccine potential. We've done some initial work in HBV. I truly believe that this, with a capsid inhibitor and a nuke, could be a functional cure for HBV. Our goal is to learn enough about the safety, show some demonstration of efficacy, and then find a partner, find a partner in the antiviral space or in the vaccine space to do the development. We're focused. We're a small company. We're focused on the immuno-oncology asset. That's what we're going to do. But we know we have the opportunity to license this to other players in other fields. Again, we have a platform. We're working on some other strains. We did announce that we plan to nominate another IND candidate later this year, probably either in oncology or infectious disease. And the last point I want to leave you with, as we're coming on the 30-minute mark, is this is an experienced management team. It's not the first rodeo for anyone on the team. And for that matter, it's not the first rodeo for anyone on our board. This is a team that's really designed to execute on the trial that we're doing and develop this type of novel therapy. In summary, if you look at the investor highlights, we are, every month we announced the key milestone last week. We have 20 patients dosed in the weekly cohort, and that's progressing. We received authorization to expand the trial in Canada. A really big thing for us was in November, getting the Decoy technology that in the Frontiers in Immunology, which is a pretty prestigious peer-reviewed journal. Our supply agreement with Beijing in October. All of this really speaks to the execution. I can tell you that I believe I'm drinking the Kool-Aid when it comes to the Decoy Therapeutics. I can't tell you what the results will be at the end of this year. I will tell you that we will execute and do our best to get those results. With that said, this is just the last slide highlighting our experienced board. We have people like Dr. Mark Gilbert, who got one of the first CAR T therapies, Dr. Martel, who's a translational practicing immuno-oncologist, to folks like Dr. Pomerantz, who actually led the first microbiome series. He was the founding CEO. That, again, is a bacteria-based, though it's a gut bacteria versus systemic. But, we have a lot of experience around the board and around the management team. With that, I just want to thank everybody. I want to thank John for the opportunity. And John, I'm open for any questions you want to bring up.
Yeah, thanks again, Jeffrey, for presenting today. Just a reminder that you can try to get in maybe a couple questions here through the Zoom chat feature, and we'll do our best to try to get those addressed. I do have a couple questions. One is in regards to your combination therapy trial. Can you expand upon that? What we should invest, how should investors look at that? What do they expect? And then, one of the questions that's related to that is that you're working with Beijing. Its symbol is ONC, and it's nearly a $29 billion company today. Well, why are they working with Indaptus in this case? And could you speak to, that was a lot of questions, but in one, but can you go try to tackle that?
Sure. Let me do the Beijing one first, since that's kind of short and sweet. Beijing has this anti-PD-1, and the number one anti-PD-1 is actually number one drug in the world, therapy in the world, called Keytruda. Merck sold almost 30 billion last year alone. So what Beijing is looking at doing is like, well, how can we get into this market? How can we change? You're not going to take on Merck and at 30 billion and knock it down, just by marketing. You got to have some sort of hook. They believe that, as we do, is they want to see how this data looks, but in a combination setting, maybe this is the way they become the next front end PD-1. So that's really why they entered the supply agreement with us and are working with us to do this trial. Now, if I got your first question right, it's really, as an investor, how to frame what should we see in this trial. So we are really getting ready to start this trial. It will go slow. What we've done in every cohort, and the combination weekly cohort will be no different, is the first patient is dosed. We really watch it. The first patients in the single dose studies were watched 28 days. So we have this long period. We adjust as we see things, and then we shrink it down as we get more patients. That's kind of the overall view. So we'll start this study shortly. It takes a while to screen and find the right patient and all the logistics. We'll start the study. We'll get, one or two patients in the first couple months or so. And then from there, if all goes well, and if they're safely tolerating the combination of a Decoy therapeutic and an anti-PD-1, then we'll start ramping it up. Now, we want to do 20 to 30 patients. And the goal there, number one goal, is measuring safety. That's the real crust of everything we've been doing. But at this point, if we do 20 to 30 patients, we should see activity with a PD-1 Decoy combo. If not, we're missing something. We either didn't get the dosing right, we missed something in that process. When you're looking for a signal like this in an open study, this is not a like placebo-controlled trial. We will get that in real time, so that we dose a patient, we wait roughly 8 weeks. We're looking, another 4-8 weeks later, we're looking to see these scans to see if the tumors are growing or shrinking. If it works really well and we're getting a high response rate earlier in the period, we'll be able to publish and say, hey, we've dosed, however many, 8, 10 patients, and we've seen, I don't know what the number is. We'll see 0 or 2 or 4, I don't know. But we'll be able to do that as the year goes on. So by the end of the year or first quarter next year, we'll have 20 patients roughly, and then we'll have, that'll be the review. If it's only, and this is what I tell everybody, if it's only one patient, the statistics are such that I don't know if that's really a meaningful signal. We need to see more than a patient. Depending upon the N, is it 2, 3, 4? If I see 4 patients out of 20 are responding well to this, in a safety study that's not designed to look at efficacy, I'm in heaven. I think that's a great outcome. If we get more, the one thing that I don't know is you could get the first 10 patients a zero response, and then patient 11, 12, 13, all are responders. And then all of a sudden you go from being, wait a second, does this work, to, oh my God, this is wonderful. That's the way these trials work. And so I can't tell you when I will absolutely know thumbs up or thumbs down. But I'm going to have some insight as we get these patients. And as we get them, we'll read out and let everybody know. So that gives you an idea of the timing and what we're looking for.
Okay, thanks, Jeffrey. By the way, here's a quick and easy one. One of the Tribe members is asking, where do we find this presentation that you presented today?
This is on our website. On our website is our corporate presentation, but if you go to there's a tab for the science. I forget what it's exactly called, but there are more in-depth presentations, and there are the posters from ASCO, SITC, AACR this year, last year, the year before. And between all of that, you get everything of the company. I mean, that's really a whole bunch. And there are some presentations on there, maybe under events and presentation, where our chief science officer, he's presented to the natural killers, the Sting agonist conference. These are all kind of communities, scientific communities, where experts in these individual technologies present. And Michael, in the last year or so, Michael Newman, who invented the technology and is our chief scientific officer, Michael's being asked pretty regularly now to attend these conferences and present on what we're doing.
Okay, thank you, Jeffrey. The other question is in regards to the stock. You recently reported, and you had, I believe, some updates of coverage. Can you speak to the analysts that are following you and how they see it?
Sure. We have two analysts that are covering us, one at H.C. Wainwright, one at Maxim. I believe the H.C. Wainwright analyst has it's an $8 target price, and the Maxim analyst has a $5 target price. I think both of them have been pretty consistent for the last couple of years, which is, the company executes, and I'm always proud. I'm an operator. We hit our goals for the most part, and that we've really hit everything we wanted to see in each of the earlier cohorts. But it really all comes down to this combination study. We either, you hate to say it, we are in biotech, and a lot of things are binary. This will be very binary for us as a company. How this combination study works with BeiGene's tislelizumab.
Okay, thank you. And then a quick follow-up there is assuming you move into a phase 2 study, would it be double-blinded?
No. The way you do these phase 2 studies is not blinded. You do what's called a controlled study. So what you do is you basically recruit patients into your study, and some patients will get, let's say we're doing a 2-arm study with tislelizumab and then tislelizumab plus Decoy 20. So the patients will know, am I in the tislelizumab only or the tislelizumab plus Decoy 20? But it's not a placebo or blinded study. It's a study that has these 2 arms in it. Because look, the patients, you need to be able to use the same sites, the same patient population, and just try one being your drug, one being without your drug. Did that make sense?
Yes.
Okay.
Absolutely. Thank you. The next question is regards to your patent portfolio, where you made some news recently about expanding it in China, Japan, and Israel. Can you speak to your patent portfolio in general and with regards to this expansion?
So the expansion was, we got our patents for this technology being used for antivirals. So now the global portfolio for the antiviral patent estate, and we have multiple patent estates, The core patents are around the use of gram-negative bacteria with LPS reduction, not LPS elimination, just reduction, and it's all in the ratios. Those patents are what we have them for the use in cancer, and then we also have them now for antivirals, and we have them in all the usual large countries in Europe and around the world.
Yeah, thank you. Just a reminder here, this week is a fairly busy week for Indaptus, business-wise, fundamentally moving their progress and their trials forward. But additionally, I'm going to be meeting Jeffrey out in Scottsdale tomorrow. I actually see him tonight, but we're hosting tomorrow at 11:30 Pacific, a luncheon event where he's going to be going to more depth and be meeting with local tribe members there. And then tomorrow night we'll be in Costa Mesa, the Newport Beach, Orange County area, and hosting a dinner event tomorrow night at 6 p.m. And then we'll be in the Orange County area for the next couple of days, and then hosting to a big new crowd in San Francisco, my hometown, on Friday this week. And so if you know anyone that if you're on this call that might want to meet Jeffrey, just ping me and let me know at john@tribepublic.com, and I'll do my best to accommodate any interest that they might have. Again, a reminder, we're going to have this event video at the Tribe Public YouTube channel later today. And again, look to get Jeffrey out across the world as he continues to make progress, and with the milestones ahead that are quite exciting, and hopefully in concert be across the U.S. with him. If you want him back on as he reports progress, remember the wish list is at your disposal at tribepublic.com. Just go there, ping us there, or again, if you want to ping me directly at john@tribepublic.com, please do so. That's JOHN@tribepublic.com. Jeffrey, I want to thank you for joining us today, and all the tribe members that joined us, and really appreciate you helping us understand Indaptus. And remember all that the company is publicly traded on the NASDAQ. The symbol is INDP. Again, Jeffrey, thanks so much, and I look forward to having you back on the tribe and seeing you again this week.
Thank you, and thank you, everybody. Have a great day.
All the best.